So we started with cell division, and in cell division, we saw that there were two types of cells. Cells can either be somatic cells, which are body cells that are not concerned with the process of reproduction, or cells can be reproductive cells, which are also called as germinal cells. There were two types of cell divisions. One is mitosis, the other is meiosis. Quickly revising, mitosis produces two daughter cells. Mitosis is your equational division because the chromosome number of the daughter cell is equal to the parent cell. Mitosis is something that takes place in somatic cells. Mitosis is something that helps in the process of growth. Mitosis is something that helps in the process of repair. Mitosis is something that helps in the process of healing and reproduction, but in lower organisms. Meiosis is your type of cell division which produces four daughter cells, is a reductional division because the chromosome number of the daughter cell is half of the parent cell. It helps in the process of production of gametes. These are the major differences between mitosis and meiosis, of which they will very commonly try and test the number of daughter cells produced and the chromosome number of the daughter cell. The number of daughter cells produced in mitosis is 2, in meiosis is 4. And mitosis may chromosome number of the daughter cell will be equal to the parent cell. Meiosis may it will be half of the parent cell. Then your animal cell diagram for identification, you should avoid looking at the cell wall and try and look for the centrosome. The centrosome, you know, is made up of centriole and aster. It may be in this form, one dot, one line that we are seeing, or it may be two dots that are present, or you may see cylinder-like structures like this that are present, and they'll be surrounded by these ray-like structures, which are called as asters. The moment you see these, you know that it is an animal cell because the centrosome, which is located in the cytoplasm of the animal cell near the nucleus, is exclusive to the animal cell. It's only in the animal cell. Outermost covering of the animal cell, you can see over here, is the cell membrane. Had it been a plant cell, that would have been the cell wall. Dense body inside the nucleus, this is your nucleolus, which disappears during your prophase, and so does your nuclear membrane. The asters, if you remember, is an MCQ question, is something that forms the spindle fibers during the process of cell division. Then you move to chromatin fibers, TIN. Chromatin fibers are thread-like structures in the nucleus, which during the process of cell division either thicken or condense. When they thicken or condense, they form structures which are called chromosomes. So this line that we have drawn, this bar-like structure that is there, that's your chromosome. And on this chromosome, the horizontal lines that you were seeing, those horizontal lines, they are the genes. So where are the genes present? The genes are on the chromosomes. First, what are chromatin fibers? They are thread-like structures in the nucleus. What happens to these structures? These structures basically thicken or condense during the process of cell division to form chromosomes. Then you remember the board question from that? The cell component which is seen only during cell division. That's a board question. Answer is chromosome. Cell component seen only during the process of cell division. So these chromosomes are basically carriers of heredity. What do they carry? As we had discussed, they're like postman. A postman carries letters. Similarly, the chromosomes carry something. What do they carry? They carry genes. What are genes? Genes are units of heredity. What are genes made up of? So genes are made up of DNA, deoxyribonucleic acid. Look at the spelling, don't make an error, NUCLEIC. What is DNA? DNA is the chemical basis of life. So once again, thread-like structures in the nucleus are called chromatin fibers. They thicken or condense during cell division to form chromosomes, which are carriers of heredity, which carry genes that are units of heredity made of DNA. DNA is a substance which constitutes the genes. Full form is deoxyribonucleic acid. Marked important 2 questions. Cell component seen only during cell division, chromosome. What makes up the genes? DNA. Full form deoxyribonucleic acid. You can see the centrosome as we had discussed, cytoplasm of the animal cell. It's only in the animal cell. It's near the nucleus. Don't write above, below, beside, anywhere. Just look at near the nucleus. It initiates and regulates cell division in the animal cell. You can see, as we had discussed, a dot and a line, or two dots and dot, two cylinders basically, and then asters forming spindle fibers during cell division. If they speak about a protein, you may see something called as tubulin that would be associated with it. Then we had drawn a duplicated chromosome, and we remembered that it would look like a ladder. The vertical lines that are there are called chromatids, and they're joined at this point, which are called as centromere. So this entire one vertical line, the one that I'm shading now right now, this entire red thing is one chromatid, and the entire white thing is another chromatid. So a duplicated chromosome actually contains how many chromatids? It contains 2 chromatids. And these chromatids are held together at this point, which is called as the centromere. Centromere is the point of attachment of sister chromatids. Why do you call them as sister chromatids? Because they belong to the same chromosome. Chromatids which belong to the same chromosome are sister chromatids, and those which belong to different chromosomes are non-sister chromatids. Where will we use this? We will use this during our discussion on crossing over. Crossing over ke definition mein we could speak about the exchange of chromatids, or the exchange of genetic segments, between non-sister chromatids of homologous chromosomes. And the point of crossing over was the chiasma or the chiasmata. And the significance of crossing over was it produces variations. Then we saw that any time that cell division takes place, there will be 2 major events. One will be division of nucleus, which is karyokinesis. The other will be division of cytoplasm, which is cytokinesis. Cytokinesis, if you remember, takes place in animal cells and in plant cells in different ways. Animal cell mein hoga, to it will be furrow formation. And plant cell mein hoga, to it will be cell plate formation. Karyokinesis is all the events involved in the division of nucleus. Okay, all the events which are involved in the division of nucleus. So when we see karyokinesis, it will be your prophase, metaphase, anaphase, telophase. All of it would be under karyokinesis. Okay. Again, then the distinguishing, you see if a cell, now we're seeing mitosis, if a cell having chromosome number X divides, it produces 2 daughter cells. So this is one daughter cell, this is the other daughter cell. If it had chromosome number X, then the daughter cells will also have chromosome number X. This is so because during synthesis phase, the DNA duplicates. So actually ये X का X plus X हो जाता है, और फिर एक X का set एक cell में जाता है, और दूसरे X का set दूसरे cell में जाता है. So mitosis is a type of cell division in which 2 daughter cells are produced, which is equational because the chromosome number of the daughter cell is equal to the parent cell, which occurs in somatic cells which are not concerned with reproduction, helping in growth, replacement, repair, reproduction in lower organism. If in case of plants, they ask you कि meristematic tissue में क्या होता है, तो meristematic tissue in plants basically is concerned with growth. या फिर they can ask you type of division at the tip of the root, या फिर the cells at the tip of the shoot, what kind of division they show. So all of that is mitosis. Then you move to meiosis. You see the cell has chromosome number X. It produces 1, 2, 3, 4 daughter cells. And all those daughter cells that it produces have chromosome number half of the parent cell. So X by 2, X by 2, X by 2, X by 2. So if a human chromosome, which normally has 46 chromosomes, undergoes mitosis, the daughter cells have 46 chromosomes. If a human cell which has 46 chromosomes undergoes meiosis, the daughter cells produce 23. The daughter cells produced have 23 chromosomes. So remember mitosis 2, meiosis 4, mitosis equation, meiosis reductional, mitosis somatic cells, meiosis reproductive cells, mitosis growth, repair, healing, reproduction in lower organisms. Meiosis gamete formation, testes में sperm formation के लिए, ovaries में ovum formation के लिए, anther में pollen grain formation के लिए, ovule formation के लिए. Then we spoke about why are gametes produced by meiosis, why can they not be produced by mitosis? Because if you see, meiosis reduces the chromosome number to half in the daughter cell. And that reduction to half is important because at some point there is going to be union by fertilization. So we've spoken about how if sperms are produced by mitosis, तो chromosome number 46 है, तो daughter cell का chromosome number भी 46 होगा, तो sperm का 46 and ovum का 46, जब fertilize होगा तो it will form 92. But it happens by the process of meiosis, so it becomes half basically. So sperm becomes 23, ovum becomes 23, they fuse to form 46. So the chromosome number is restored during fertilization. And that fertilization has to happen. That is why meiosis takes place to reduce the chromosome number to half. So gametes कैसे बनता है? Gametes meiosis से बनता है. Then we saw mitosis में karyokinesis के phases pro, meta, ana, telo. Remembered them by P, M, A, T. Pro was pehla, meta was middle, ana was adha and away. Remember anaphase we had written adha and away. Adha because the centromere divides and the chromatids move to opposite poles. And telo we had spoken about being opposite of pro. Then we drawn the animal cell diagram, a typical animal cell, the same that we had seen. And we'd seen what were the changes in prophase. So if you observe the diagram, a diagram-based question, be sure to look for whether you see a disappearing nuclear membrane. That's going to be your key to understanding if it is prophase. Then look for the aster and see if you can see the asters. So if you can see the asters, you can see the centrioles, then it's an animal cell. So it's an animal cell in prophase. Then count the chromosomes. So this is 1, this is 2, this is 3, this is 4. Does that mean that there will always be 4 chromosomes? No. There could be 6 also. You just have to count the number of centromeres. If you have a doubt, then count the number of centromeres in prophase. Should not be a problem. This is 1 chromosome, this is another chromosome. Okay. So prophase में 2 structures disappear होते हैं. पहला होता है nuclear membrane, दूसरा होता है nucleolus. एक structure thicken होता है. ये जो chromatin fiber होता है, ये thicken करके बनाता है chromosome, and this is a duplicated chromosome. एक structure divide होता है. The centrioles duplicate and they duplicate and they move to the opposite poles. And the spindle fiber formation will begin. That's what happens in prophase. Nuclear membrane and nucleolus disappears. The chromatin fiber condenses, it forms chromosomes, the centrosome duplicates, the centrioles move to the opposite poles and spindle fibers appear. So spindle fibers appear in prophase, spindle fibers disappear in telophase. Nuclear membrane, nucleolus disappear in prophase, nuclear membrane, nucleolus reappear in telophase. Chromatin condenses to form chromosome in prophase, chromosomes uncoil to form chromatin fibers in telophase. The same thing we've done, then we've spoken about how we draw a plant cell. We've drawn a plant cell. In the same manner, we added a cell wall. We changed the shape. Need not always be of the same shape though. We changed the shape, we added a cell wall and we removed the centriole. And we did a prophase in the plant cell also. Again, easy to identify because what you're looking for is the disappearing nuclear membrane. And you get that this one over here, this entire dotted line that you are seeing, that's the disappearing nuclear membrane and no centrioles here or no centrosome present over here. So this is a prophase. Following prophase, everything else remains the same with obviously no reference to the centrosome and the centriole. Then goes metaphase, M meta, M middle. All the chromosomes that are there, they align at the equator. So you will see all of them aligned at one straight line. Need not be exactly a straight line. You may have a little zigzag also, but as long as you're seeing everything centered in the middle, it is metaphase. One of the most easiest ones to identify also, substrate line mein dikhta hai. It could be like this or it could be like this. If it's like this, then you will have your centrioles over here at the poles. And if it's like this, you can see this is the centrosome. So this dot and line is the centriole. The ray-like structures are the asters. Here, there is a special additional labeling that we put, which is the achromatic spindle fiber. The achromatic spindle fiber is the fiber which does not pass through any centromere. So there's a spindle fiber that's not passing through any centromere. It's your achromatic spindle fiber. Specialty chromosomes align at the equatorial plane. The same thing happens in the plant cell also. Then you move to your anaphase. Anaphase, I told you, adha and away. So the chromosome, which was basically like this, you can see over here, this was the centromere to which the spindle fiber was attached. This was your spindle fiber. The spindle fiber will contract. When the spindle fiber will contract, the centromere will divide and this part will be pulled and this part will be pulled. You can see this one chromosome converts into this structure. So what happens, and you can see they move to the opposite poles. So the centromere divides and the chromatids move to opposite poles due to contraction of spindle fibers is what happens in anaphase. And you can see a slight furrow beginning to form in the animal cell, late anaphase, but we do not study early and late, so we need not worry about it. We just study prophase, metaphase, anaphase, telophase. So you see that small furrow beginning, that small furrow that is beginning is the beginning of cytokinesis or the beginning of cytoplasm ka division in the cell. You can see the changes. So what happens? Centromere divides. Sister chromatids move to opposite poles. Why? Because the spindle fiber contracts, and during late anaphase, here you will see V-shaped, L-shaped, J-shaped, I-shaped chromosomes. V-shaped. So you can see this is the V-shaped chromosome. Okay, now the shape of the chromosome depends on the location of the centromere, but we don't have that, so we need not worry. But if they ask you V-shaped, L-shaped, J-shaped, I-shaped chromosomes, that will be seen during the anaphase stage. After anaphase comes same thing in animal cell also, in plant cell also. Then we move on to telophase. So if you see, telophase was the opposite of prophase, and you can see how much this furrow has deepened over here. And the nuclear membrane has reappeared, which was not there till now. So you will see that there are two nuclear membranes, two formed cells ready to divide and then grow. This is telophase, opposite of prophase. As we had discussed, nuclear membrane, nucleolus reappears. Cytoplasmic furrow, या फिर cytoplasmic constriction deepens. Spindle fibers disappear. Followed by that, you will find two daughter cells that are produced, and since it was mitosis, it was identical daughter cells. Same thing in plant cell, but with the difference being that of the cell plate formation that you were seeing. This is the cell plate. The cell plate is formed at the equator. The furrow formation that was there was taking place from outside to inside. We call it as centripetal. The cell plate is growing from inside to outside. It is centrifugal, not asked frequently, but just good to know. Animal cell में cytokinesis cleavage furrow formation से होता है. Plant cell में cell plate formation से होता है. Animal cell में centripetal direction में होता है. Plant cell में centrifugal direction में होता है. Then we saw this as a board question. So if you see this as a board question, you will see anaphase. Yes, which are listening.
Sir, can you repeat the centripetal and centrifugal?
Centripetal is moving from outside towards the center, from outside towards the middle, and centrifugal is moving from the middle towards the outside.
So which is the most common one?
Okay, this depends on which cell it is, no? Animal cell or plant cell. If it is animal cell, then furrow formation is centripetal. If it's plant cell, then it is cell plate formation, which is always centrifugal.
Okay.
Sir,
That extra terms, need not worry about them. Listening?
sir, in prophase, do all the chromosomes have spindle fiber passing through them?
Not required. You just have to see a disappearing nuclear membrane from that.
So could you explain a haploid and diploid number?
I will take all your doubts at the end of the session. Okay? So given below is a diagram, you can see this one is anaphase. It is very clearly identified because you can see your V-shaped chromosomes. If they ask you the chromosome number of this cell, okay, if they ask you the chromosome number of this cell. So here the chromosome number is 1, 2, 3, 4. This is a chromatid which is formed from this one chromosome. So chromosome number is still 4 over here. Because this is anaphase we identified. It's an animal cell we identified because we can see the centriole and the aster that is present over there. That's the anaphase that is, that's the anaphase that we are looking at. And we can see the centriole and the aster. The one that follows this, the one that follows anaphase we know is telophase. So in telophase, we can show 4 in difference. So that's how it is solved. I don't think that should be a problem for you. This one is a telophase diagram. This is also a board question which we had done. You can see the cytoplasmic constriction. You can see the nuclear membrane disappearing. So some of you might have thought of it as prophase, but prophase you will not have these two daughter cell kind of things there. No, that's only an identification feature of telophase. You can count the number. You can see 1, 2, 3, 4, 5, 6 chromosomes that are present. And they had asked you to draw the stage that comes before telophase. So anaphase, see here, anaphase में 6 chromosomes. So this plus this is 1. This plus this is 2, this is 3, this plus this is 4, this plus this is 5, this plus this is 6. Okay, so anaphase mein, if you have to count the two chromatids and join them as one chromosome. That's the place where many of you make a mistake. This you can see is a disappearing nuclear membrane. A disappearing nuclear membrane will be seen during prophase. So it's prophase. Then they had asked in what 2 ways is mitotic division, the third one. In what 2 ways is the mitotic division in animal cell different from the mitotic division in plant cell? So mitosis in animal cell obviously involves the centrosome. Plant cell does not involve the centrosome. mitosis in animal cell cytoplasmic division occurs by furrow formation, and plant cell cytoplasmic division occurs by cell plate formation. This particular year, they had asked, name the type of cell division which occurs in the growth of the shoot. So growth of shoot is mitosis. And pollen grain formation. So pollen grain is the male reproductive cell in the plant. So pollen grain formation was meiosis. Then you move down, you've done one more. This we had done. So this was another one. Again, the identification feature for the stage was the disappearing nuclear membrane. So it was prophase, but you did not see the centrioles and the asters. So it was plant cell. So it was prophase in plant cell. That's how we had identified it. Prophase in the plant cell. Then we went on to seeing the cell cycle. We said just like you have a life cycle, similarly the cell has a cell cycle. Okay, from the time the cell is born till the time the cell divides again. So it's after cytokinesis that the cell is born, and it is after G2 phase that the cell divides, and this entire thing. So if you see the process of growth of the cell along with the division of the cell, so growth along with division, then that constitutes your cell cycle. But if you only see growth, which is G1, S, and G2, then that constitutes your interphase, which is the time period between two successive divisions. Only the growth part is interphase. Interphase is misnomerly called as the resting stage, they say, no? Interiorly, on the inside rather, it is dividing, it is very active, it is just preparing for the next cell division. So growth plus division, cell cycle. Between two successive cell divisions, interphase. Interphase ke 3 stages, G1, S, G2. S phase is the important one. There's already an MCQ that has been asked on S phase as a board exam. This duplication of DNA takes place. So DNA was the answer there. The duplication of DNA takes place in the S phase. Then we done our logical sequences. Cell cycle. Here I'd put cell cycle is basically the cell division plus the cell growth. K plus C is karyokinesis and cytokinesis, and cell growth is interphase. Karan, at the end of it, I will take you down. Interphase, you can see, is time period between two successive cell divisions or end of one cell division to start of next cell division. So that's what we had seen. Now within the logical sequence, again, some very commonly asked board questions are on logical sequences from the cell cycle, G1, S, G2, pro, meta, ana, telo, cyto. This G1, S, G2 can be written as interphase. The pro, meta, ana, telo can be written as karyokinesis. So you could have either of it, and you should remember to put it in place. Interphase G1 mein, yes, G1 mein, G1 is, as I told you guys to remember it as G1, the cell first gets life. So G1, first growth phase. After, just after mitosis, what happens? The cell basically increases in size. The chloroplast and the mitochondria, which have their own DNA, they duplicate. They duplicate by fission. And RNA and proteins are synthesized. Now, you may have seen certain MCQ questions which say RNA and proteins are synthesized in which phase. And they may have given you what? They may have given you the idea of G1 phase as well as G2 phase. Okay. Ideally, RNA and proteins are synthesized in both. So you need not worry, they will not give you overlapping options. And if they do give you overlapping options, you will have more than one correct. So don't stress about these questions which are framed incorrectly. Okay. So post-mitotic phase, cytoplasm increases in size, cell sizes increases, mitochondria, chloroplast, which have their own DNA, they duplicate. Now from here, the cell may either leave or the cell will enter into the next phase. When it leaves, it is the R phase. Some people call it as the G0 phase also. They differentiate and they do not divide. Differentiate is when a cell assumes a specific shape to perform a function. S phase is basically your synthesis phase, in which what happens? The DNA duplicates. Okay, DNA duplicates in S phase. This is the most important part here, which is where the MCQs are asked from. So the DNA doubles or the DNA duplicates or the chromosomes duplicate, whatever you see. But it is the DNA, remember, that we are looking at. G2 phase, RNA and protein synthesized, which are basically required for the next cell division. Then we went to seeing meiosis, and we saw that there were two stages in meiosis, meiosis 1, meiosis 2, which we did not have to learn. But for our reference, we saw that meiosis 1 is actually the reductional stage. And then meiosis 2 hota hai, in which two daughter cells are produced. Meiosis 2 is actually nothing but mitosis. It's similar to mitosis. In meiosis, apart from the things that we had already covered, we had to do the process of crossing over, in which we saw that it was the exchange of genetic segments between non-sister chromatids of homologous chromosomes. And the definition of homologous chromosomes, chromosomes of equals one from each parent, equal size and shape. Sometimes they may add bearing the same genes. Corresponding same size, shape, one from each parent. This was in the sample paper also. The point of crossing over is chiasma or chiasmata. It leads to genetic recombinations, or it leads to variations. Variations are differences among organisms. So one of the causes of differences is variation. Another cause of variation could be mutation also. Mutation is the sudden change in one or more genes or in the number or structure of chromosomes. Mutation was also heritable. So this was the definition that we had seen, exchange of chromatid material. This was your textbook definition, exchange of chromatid material between the maternal and paternal chromosomes, one which comes from the mother and the other which comes from the father. That's what constitutes the homologous pair. The point of crossing over, chiasma, significance, produces genetic recombinations or produces variations. Then the second part, rather the first part, was the common question that is asked from that part is naming the 4 nitrogenous bases and what combines with what and how do they combine and what is a nucleotide basically. So we had spoken about the structure of the chromosome. When we opened up a chromosome, we found a chromatid. When we open up the chromatid, we found the chromatin fiber. When we opened up the chromatin fiber, we found repeating units called nucleosomes, which were bead-like structures. So these nucleosomes that were found, these nucleosomes were basically a core of 8 histone proteins. Here we put up an image, you can see 1, 2, 3, 4, 5, 6, 7, 8. So 8 histone proteins core on which a strand of DNA wraps around. That's what a nucleosome is. So you can see multiple nucleosomes. Remember, it's a core of 8 histone proteins. So histone is basically the protein that is associated with the compaction of the chromatin material or the packaging of the DNA, if you may call it in that way. Then if you open up the nucleosome ka DNA, then you will see on the DNA structure, this over here was your nucleotide. Your nucleotide is a complex made up of base, sugar, phosphate. How was the base? The base was nitrogenous. The phosphate, and how was the sugar? The sugar is pentose, matlab 5 carbon wala. And in this case, the sugar is deoxyribose, because it's DNA, no? So deoxyribose wala sugar. How many strands do you see? 2 strands are basically the vertical lines. What is the shape? Double helix. There's a central imaginary axis around which it is coiled. The double helix model of the DNA given by Watson and Crick. It says polynucleotide chain because there are multiple nucleotides. See, this is one nucleotide. This is another. This is the 3rd nucleotide. This is the 4th nucleotide, the 5th nucleotide, the 6th nucleotide. We have seen questions only on nucleotides, so you should know how to identify. This is your nucleotide. On the inside will always be the nitrogenous base. On the outside will always be the phosphate. The nitrogenous bases are of 2 types, purines and pyrimidines, but that's not in syllabus. We just need to know the names. The names of the 4 nitrogenous bases, the adenine, the guanine, the cytosine, and the thiamine. This is very important, and look for the spelling. They will try and confuse with thiamine ka spelling. thiamine, that is vitamin B1. This is thyamine. So adenine pairs with a thiamine with a double hydrogen bond. A with T is with a double hydrogen bond. And guanine pairs with a cytosine. That's a triple hydrogen bond that is present. What is a nucleotide? A nucleotide is a complex made of 3 things. Remember, we've spoken about base, sugar, and phosphate. Then we wrote down the 4 nitrogenous bases, the adenine, the guanine, the cytosine, the thiamine. Then we drew nucleotides, identifying them. And you know, successive nucleotides are also linked like this. So this is how successive nucleotides are also linked. The hydrogen bond in between, we can see double hydrogen bond, triple hydrogen bond. And we spoke about Aya, Tha, Chala, Gaya. A for Aya, Tha ka T, Chala ka C, Gaya ka G. A with T, double hydrogen bond. C with G, triple hydrogen bond. What makes up the rungs of the DNA ladder? So the rungs of the DNA ladder are basically made by the nitrogenous bases and the hydrogen bonds present in between. Phosphate is always directed to the outside and the nitrogenous bases are directed to the inside. Then the last thing was the definition of genes, which are specific sequences of nucleotides. We've seen MCQs with only this much asked. Specific sequences of nucleotides. Specific sequences of nucleotides are genes. What do genes do for? Genes basically code for proteins. And what do the proteins do? They express themselves as features of the body. Okay, so that was what we had to do in cell division. Now we can take your doubts on cell division. Fine, let's move to genetics. So genetics, a lot of things are cancelled in genetics. We will see what is required for us. So genetics is basically the study of heredity and variation. What is heredity? Transmission of genetically based characteristics from parent to offspring. Where we had discussed of the various examples, if you lose your hand in an accident, then your child will not be born without a hand. Not genetically based, it's something that's related to the somatic cells. So it has to be genetically based characteristic, only then can it be transferred from one generation to another. Variations we spoke about were differences between sexually reproducing organisms. Then we went on to character and trait. So what is a character? Any inheritable feature of an organism is called as a character. Things which get transferred from one generation to another, like height, for example, is a character, or color of your eye is a character, or shape of your hair or your eyebrows, or color vision or tongue rolling. These are characters in which you could remember color of eye, or you could remember color vision and tongue rolling. And they could exist in different forms. So the different forms in which these characters exist were called as traits. So we spoke about characters and traits, where character is the inheritable feature of an organism and traits were the alternative forms of the character. In relation to the pea plant, if height is a character, then the plant can be tall or dwarf. So tall and dwarf are traits. Father of genetics, Gregor Mendel. Choice of material, garden pea plant. Biological name of the garden pea plant, Pisum sativum. P capital, S chota, underlined separately. Make sure you get the spelling correct. So Pisum sativum. Then we moved on to reasons for selecting the pea plants. There were many varieties available. Pure forms were there, naturally self-pollinated, easily cross-pollinated. So then the table, there's an important table which speaks about the different characters and the traits. They can give you a list and they can ask you which of the following was not what Mendel selected. So height of the plant can be tall or the plant can be dwarf. Tall is the dominant one. Dominant were represented by capital alphabets. So we see dominant can be represented in two ways, capital or capital small, you remember, capital or capital small. And he also said that the capital will always express itself. So if there is one capital T and the other is small, still the plant is going to be tall. If both are tall, then obviously it's going to be tall only. But this small t will only express itself in this condition, which means dominant will always express in homozygous as well as heterozygous, but recessive will express only in homozygous. That's the MCQ that we've been seeing during the rounds, that a dominant gene expresses itself in homozygous and heterozygous condition, while a recessive gene only expresses in the homozygous condition. Dominant expresses in homozygous and heterozygous condition, recessive expresses only in homozygous condition. Then we saw the position of the flower could be axial and terminal, where axial is basically dominant, terminal is recessive. So axial and terminal we'd seen that axial is this angle that it makes here. Inflated, constricted, green and yellow, round and wrinkled. And I told you they can ask you between this. Pod color mein dominant is green and seed color mein dominant is yellow. This is the different one actually, because normally when you think of a pea seed, you would think of green, but seed color mein dominant is yellow and pod color mein dominant is green. Then we went on to seeing his process, and we saw he took all tall plants, he took pure tall plants, crossed them with pure dwarf plants. The first generation, all the plants were tall, which basically made us understand that tall is something that is dominant. And that's one thing that you can use for your Mendel's law of dominance. Out of a pair of contrasting characters present together, only one is able to express itself. The one that expresses itself is dominant, the other is recessive. Make sure to learn all your laws. There are 3 laws. They can come and you have to learn the statement so you can easily distinguish. F1 generation se law pucha, और monohybrid cross है तो you can write the law of dominance. When he crossed the F1 hybrids, he found that 3/4 were tall and 1/4 were dwarf. That is how we came about the 3:1 ratio and the 1:2:1 ka ratio. So if you see here, we had done tall hai, so tall can be represented as capital T small t or capital T. But pure tall hai, pure matlab homozygous, matlab both the alphabets had to be the same. So capital T separated them. Pure dwarf है, dwarf लिखने का एक ही तरीका है. Dwarf can only be represented in this way that you are seeing over here. So dwarf can only be represented in this way, small t, and you separate them. Remember, I'll just do one, capital T, small t. We separated capital over here, small over here, and small over here. And then we put them in the box that is present over here. We put capital T here, small t here, and small t here, and we just cross multiplied it. And we found our first Punnett square of the F1 generation. And we found that all the plants in the F1 generation were basically tall, okay? Capital T and small t. So this was the capital there. So obviously it was going to be tall. So all plants produced were tall. F1 generation में they don't ask you ratios क्योंकि सारे same है. But they ask you genotype, phenotype. So you should know genotype is writing the spelling and phenotype is writing the expanded form. ये capital T small t जो हमने लिखा है ये इसका genotype है. ये plant दिखेगा कैसा वो उसका phenotype होगा. Remember, genotype is set of genes present and phenotype is external observable character. External observable characters are phenotype and set of genes present is the genotype that is there. So capital T small t becomes genotype. And phenotype is वो plant बाहर से कैसे दिखेगा तो capital हमेशा express करेगा तो all plants produced will be tall. Then F2 generation में we did the same thing and we found that 3/4 were tall, 1/4 were dwarf. So the phenotypic ratio was 3:1. Then when you have to write the genotypic ratio, you have to look for whether it is homozygous or whether it is heterozygous. When it is homozygous, both the alphabets that you're writing will be the same. In case you don't understand genes and alleles, both alphabets same, homozygous. Same matlab dekho, capital T, homozygous. Small t, also homozygous. Capital T, small t, heterozygous. So you found that one was pure, two were hybrid, and one was pure dwarf. So the genotypic ratio became 1:2:2. Then we did another one for practice. We saw pure axial, pure terminal. This was just for our practice. And we again did 1:2:1 and 3:1. Then what exactly is a monohybrid cross? A monohybrid cross is a cross between two individuals or between two different traits for a single character. यहाँ पे जो character consider किया आपने वो axial and terminal flowers वाला character था. मतलब it was position of flower. What is the flower position? So position of flower was the character. और उसका दो alternative traits था, two different traits. So it could either be axial, here you go, there is axial and then there is terminal. These are the traits for that character that you had chosen. Okay, so I had given you character position of flower है, कितना character है एक, उसका कितना trait है दो, axial और terminal. फिर देखा था character height of the plant, एक ही character उसका दो traits, tall and dwarf. Then we saw the law derived from the monohybrid cross and we spoke about the law of dominance. टॉल और ड्वार्फ जब मेंडल ने क्रॉस किया तो फर्स्ट जनरेशन में खाली टॉल दिखा, ड्वार्फ नहीं दिखा, और सेकंड जनरेशन में वो ड्वार्फ वापस आ गया. बट लॉ ऑफ डोमिनेंस खाली वो फर्स्ट जनरेशन के लिए कि आउट ऑफ द पेयर ऑफ कॉन्ट्रास्टिंग कैरेक्टर्स ओनली वन इज एबल टू एक्सप्रेस इटसेल्फ. तो टॉल एंड ड्वार्फ में से किसने एक्सप्रेस किया? टॉल ने एक्सप्रेस किया. द अदर रिमेन सप्रेस, दूसरा वाला एक्सप्रेस नहीं कर रहा है. सो दूसरा वाला एक्सप्रेस नहीं कर रहा है तो वो रिसेसिव है, तो ड्वार्फ रिसेसिव हो गया और टॉल डोमिनेंट हो गया. फिर जीनोटाइप फिनोटाइप का डेफिनेशन, इंटरनल जेनेटिक कॉन्स्टिट्यूशन ऑफ एन ऑर्गेनिज्म, एक्सटर्नल ऑब्जर्वेबल कैरेक्टर्स व्हिच आर जेनेटिकली कंट्रोल्ड. ये जेनेटिकली कंट्रोल्ड अंडरलाइन किया था क्योंकि किसी बोर्ड क्वेश्चन में फिल इन द ब्लैंक करके आया था, तो एंड में जेनेटिकली कंट्रोल्ड होना चाहिए. देन वी डन अनदर प्रैक्टिस वन, inflated, constricted. Again, you don't require it, it's the same thing. Then we spoke about alleles क्या होता है. So first we had actually done everything and then last में I introduced you to the allele वाला concept. So allele is nothing but a gene. Genes can exist in multiple forms. Those multiple forms of genes are called as alleles. बट जो पूछा जाता है वो डेफिनेशन होता है. So alleles are alternative forms of the gene occupying the same locus. Locus का मतलब होता है position. On which chromosome? On homologous chromosomes. Affecting the same characteristic. Which is the characteristic? Supposing you are considering capital T. Then the same characteristic would be the height of the plant. And we remember we said that we will define our allele keeping in mind heterozygous condition. तो हमको definition learn करने में easy होगा. तो capital T, small t सोच के हमने define किया था. तो heterozygous condition सोच के define करो. Alternative forms of genes on the same locus. Locus is position. कौन सा chromosome? Homologous chromosome. Affecting same characteristic, height of the plant in different ways. One is making it tall and the other is making it dwarf. फिर हमने बोला कि homozygous क्या होता है. तो capital देखो, capital R homozygous है. Both alleles are identical. Small r भी homozygous है. Alleles are identical. जब कैपिटल आर गमीट बनाएगा तो आधा गमीट कैपिटल आर होगा, दूसरा आधा भी कैपिटल आर ही होगा. मतलब इट विल प्रोड्यूस ओनली वन टाइप ऑफ गमीट. जब कैपिटल आर गमीट बनाएगा तो हाफ में कैपिटल आर होगा और हाफ में स्मॉल आर होगा. मतलब इट विल प्रोड्यूस मोर देन वन टाइप ऑफ गमीट. सो होमोजाइगस इज अ कंडीशन इन विच एलील्स आर आइडेंटिकल एंड हेटेरोजाइगस इज अ कंडीशन इन विच एलील्स आर नॉन आइडेंटिकल. सो दैट्स योर डिफरेंस बिटवीन होमोजाइगस. अच्छा, then I told you to identify. You've seen many MCQs doing the turns in this one. Capital R बोला तो दोनों alleles identical homozygous. और capital alphabet है तो dominant. Capital R small r बोला, एक capital है दूसरा small है तो heterozygous. But तो भी capital alphabet है तो dominant. Small r बोला, both alphabets are same तो homozygous. But small alphabets, both of them, so recessive. So homozygous dominant, capital R. Heterozygous dominant, capital R, small r. Homozygous recessive, small r. Heterozygous recessive होता ही नहीं है. Recessive हमेशा homozygous होता है. Heterozygous recessive क्या होगा? Capital R, small r. ये capital तो express करेगा, तो heterozygous recessive होता ही नहीं है. तो dominant, ये फिर से same MCQ that I was talking about. Dominant expresses in homozygous as well as heterozygous condition, while recessive expresses only in homozygous condition. Very important MCQ. This one is about question recessive वाला. Dihybrid नहीं है तो we are not wasting our time on it. We just have the law derived from the cross. So the law derived from the cross is the law of independent assortment. You have to learn the law. There's no escaping that. So when there are two pairs of contrasting characters, the distribution of alleles of one character into the gamete is independent of the distribution of the other, which basically meant कि ये capital R कहाँ जाएगा. That is not based on कि ये capital A कहाँ जाएगा. So basically it says independent. Independent is free of. Assortment is separating. So they separate independent of the other character or independent of the other allele. So they move by themselves. They are free to combine with what they want. In short, they are free to combine with what they want. You have to learn the definition. Then we spoke about autosomes and allosomes. Human chromosome number is 46. 46 are arranged as 23 pairs. Homologous chromosomes repeat हुआ था जो cell division में था. Pair of corresponding chromosomes says same size and shape, one from each parent. ये जो 23 pairs हैं उसमें से 22 pairs autosomes होते हैं. They are not concerned with reproduction. जो 23rd pair होता है वो allosome होता है. Allosomes determine the sex of the individual. XY होगा तो male होगा, XX होगा तो female होगा. Okay. Male produces two. We don't have that gender determination. So again, we are not doing that. XY रहेगा तो male होगा, XX रहेगा तो female होगा. Every time a child is born, there is equal possibility that the child can be male or the child can be female. Okay. Then mutation का definition: sudden heritable change in one or more genes or in the number or in the structure of chromosome. Again, a definition which has already been asked. Heritable is something that we add from our sides. Remember, mutation heritable होता है, मतलब it can be transferred to the next generation. दो examples दिए हुए हैं आपको. एक वहाँ पे sickle cell anemia का दिया हुआ है. Gene mutation जिसमें DNA changes, RBC becomes sickle shaped like this. The third one was the law of purity of gametes, which is also called as law of segregation, which holds true, does not have any exceptions. So every 1 minute. Every time you write the gametes, when you break them, as you can see over here, the two members that you were seeing of a pair of factors, they separate during gamete formation. Factor का मतलब gene होता है. Mendel used to call genes as factors. तब तक gene discover नहीं हुआ. खाली ये पता था कि कुछ हो जाता है या कुछ ट्रांसफर होता है, बट उसको जीन नहीं बोलते थे बेसिकली. So the two members of a pair of factors separate during gamete formation. They do not blend, they do not mix with each other. During gamete formation, they segregate or they separate. That's the idea of law of segregation. ठीक है, ये वाले इसमें इतना ही करना था. Dominant recessive आपको आता है. ठीक है, we take your doubts from genetics. Okay, so the third one was absorption by root. So transport in plants, basically transport can take place in two ways. It can either be passive transport or it can be active transport. So passive transport is moving from higher concentration to lower concentration. Passive transport does not require any energy. Active transport is moving from lower concentration to higher concentration. Active transport is what they ask you to define. So when there's movement that's taking place from lower to higher concentration, which is against the gradient, from lower to higher concentration against the gradient, it's something that is going to require energy. And then when it is going to require energy, then you call it as active transport. When it is requiring energy, then you will call it as active transport. So remember, active is against the concentration gradient and active is something that is going to require energy. Active transport ka significance for plants is going to be absorption of minerals. Passive transport, we're going to be two types. Two types or other two ways in which you could learn it. One of which is diffusion and the other is osmosis. All definitions in this chapter. Remember, you will have 5 definitions. Learn them properly. What are the keywords that you will look for diffusion ka definition? So diffusion ke definition mein we've underlined higher concentration, lower concentration. They may give you contact. Okay, they may give you direct contact. No energy, but osmosis mein we look for water. We will look for higher concentration, lower concentration, and we will look for semi-permeable membrane. Examples of semi-permeable membrane would be whisking bag, goat's bladder, animal bladder, any animal bladder, sheep bladder that you may see, cellophane paper that is present. All these will be examples of your semi-permeable membrane. Why do you call it semi-permeable? Because it allows only certain molecules to pass through it. Significance of diffusion will be exchange of respiratory gases. Active transport will be mineral absorption. So remember, it will be the process by which minerals are absorbed. Osmosis will be the process by which water is absorbed, and diffusion will be exchange of respiratory gases. Diffusion, may you just need to know the definition and you need to know the significance. This name the following: the process by which molecules distribute themselves evenly in the space provided. That is diffusion. A simple example of bottle of perfume opened in one corner of the room, or pollinating agents being attracted, insects being attracted because of fragrance, example of diffusion. Then we saw the experiment wherein we spoke about how osmosis takes place, and we spoke about observation dekhna hai, so you always have to look for a rise or a fall. We saw about tonicity and we saw 3 different types of solutions. Okay, we can see the examples: whisking bag, cellophane paper, cell membrane, animal bladder, egg membrane. We spoke about muslin cloth being freely permeable, rubber sheet being impermeable. अगर वो experimental setup आपको दिया हुआ है और वो आपको पूछता है कि इसके लिए कोई control है, तो वो कंट्रोल में क्या यूज कर सकते हो? तो फ्रीली परमिएबल मेम्ब्रेन यूज कर सकते हो, या इम्परमिएबल मेम्ब्रेन यूज कर सकते हो, या फिर यहाँ का सोल्यूशन और यहाँ का सोल्यूशन इक्वल यूज कर सकते हो ताकि ऑस्मोसिस नहीं हो बेसिकली. He spoke about the I put a control over here. State the control. Whisking bag could be replaced by the rubber sheet or the muslin cloth, or the same solution in the beaker and the bag. Then when we were learning, you can see there's pure water outside here, and there is 2% of sugar inside. So here inside there is 2% of sugar. There's lesser water obviously, so water will move from outside to inside. And we spoke of it with reference to the cell. The semi-permeable membrane could become your cell membrane, and you could speak about it with reference to the cell. So you will see a rise in the level of the solution in the Thistle funnel. When outside there is more water, water will move in. When inside there is more water, then water will move out. Water will basically move from its region of excess to its region of deficiency. If you're seeing a rise in the level of water, that means outside there was more water. If you're seeing a fall in the level of water in the Thistle funnel, that means inside there was more water. Okay, so Osmosis could be end osmosis or exosmosis. You remembered En was for entry. D was due to entry due to osmosis. किसी सेल के अंदर अगर पानी जाएगा तो सेल का साइज बढ़ेगा. When the size of the cell will increase. किसी सेल के अंदर पानी जाएगा तो सेल का साइज बढ़ेगा. The size of the cell will increase. So they will ask you, a cell was placed in a particular type of solution and the cell size increased. So if cell size increased, मतलब पानी अंदर गया मतलब end osmosis. End osmosis मतलब बाहर का solution hypo, because we had learned it like that कि endo will always happen when the outside is hypo, या फिर X will always happen when the outside is hyper. So endo is hypo. अगर किसी चीज के अंदर पानी जा रहा है मतलब सेल के बाहर का solution hypo है. What is the meaning of hypo? Hypo का मतलब होता है less. But what is less in a hypotonic solution? Solute is less in a hypotonic solution. What is more in a hypertonic solution? Solute is more in a hypertonic solution. तो अगर 2% of sugar solution है and 4% of sugar solution है, तो जो 4% of sugar solution वाला है, this is hyper in comparison to this, which is hypo. So hyper, hypo and iso are to be remembered in terms of solute concentration. And end osmosis, exosmosis कब होता है, you have to remember based on tonicity. Hypo बाहर है तो endo, cell size बढ़ेगा. हाइपर बाहर है तो X मतलब सेल साइज कम होगा. आइसो बाहर है तो सेम, देयर विल बी नो चेंज इन सेल साइज. ठीक है, दिस इज योर आइडिया ऑफ, yes, this is all what we did. See, 4% of sugar solution inside, 2% of sugar solution outside. 4% अंदर मतलब how much of water? 96. 2% मतलब कितना water? 98. So water will move from where to where? 98 to 96. There will be a rise in the level of water. End osmosis. 2% inside, 6% outside. 6% is 94, 2% is 98 outside. But you might have seen ऐसे कोई भी questions नहीं आते 2%, 4% पे. आपको कौन से वाले आते हैं? यहाँ पे they will put pure water and यहाँ पे they will put खाली sugar solution. तो you have to understand कि sugar solution मतलब पानी कम है तो पानी यहाँ से यहाँ जाएगा. या तो उल्टा कर देंगे. What they will do? They will place in the bag, they will place pure water and here outside they will place sugar solution. Then they will go from here to here. Achha, one thing that they have asked is dilute and concentrated wala. So dilute and concentrated wala mein, they will give you like this and they will tell you in the bag there is dilute solution. Outside there is concentrated solution. So dilute ka matlab more water. Water will always move from its excess to deficiency. So more water and outside is less water. So water will move from inside to outside. So there'll be a fall in the level of solution. So hypo is endo, hyper is X and iso is no change. These three boxes are important. सेल के बाहर का solution अगर हाइपोटोनिक है तो सेल के अंदर पानी जाएगा, एंडोस्मोसिस होगा और सेल का साइज बढ़ेगा. सेल के बाहर का solution अगर हाइपरटोनिक है तो एक्सोस्मोसिस होगा, सेल का साइज घटेगा. सेल के बाहर का solution अगर आइसोटोनिक है तो नो ऑस्मोसिस होगा, सेल का साइज विल रिमेन द सेम. ओके, द बोर्ड क्वेश्चन 2014 वाला, दे हैड गिवन सेल का साइज इंक्रीज हो रहा है मतलब सेल के अंदर पानी जा रहा है मतलब बाहर का solution हाइपोटोनिक है तो 1 इज हाइपो. सेल का साइज डिक्रीज हो रहा है मतलब सेल के बाहर पानी जा रहा है मतलब बाहर का solution हाइपरटोनिक है. सेल साइज इज रिमेनिंग द सेम मतलब नो ऑस्मोसिस मतलब आइसोटोनिक. Then cell sap of root hair is hypertonic. रूट के अंदर पानी जाता है by osmosis, मिनरल जाता है by active transport. आपके लिए फिलहाल ये कॉन्सेप्ट है. तो अगर रूट के अंदर पानी जा रहा है मतलब रूट के अंदर पानी कम है. ओके, रूट के अंदर पानी. और दूसरा वे देखने का क्या है कि रूट के अंदर पानी जा रहा है मतलब एंडोस्मोसिस हो रहा है. तो एंडोस्मोसिस कब होता है? जब बाहर का सोल्यूशन हाइपो होता है. तो अगर बाहर हाइपो है तो अंदर हाइपर है. So the root actually concentrates. What does the root do? The root concentrates. The root basically concentrates. What does it concentrate? It concentrates minerals inside. So when the root concentrates the minerals inside, when the root concentrates the minerals inside, the root becomes hypertonic, or the cell sap of the root hair becomes hypertonic. This is also a board question. Cell sap of root hair is hypertonic. एक और जगह पे hypertonic आता है. The space between the cell wall and the cell membrane in a plasmolysed cell is filled with. So cell wall and cell membrane के बीच का space in a plasmolysed cell is filled with hypertonic solution. Permeability में your cell wall is freely permeable and your cell membrane is semi-permeable. Then we move ahead. Hypo का definition देखो. Hypo is less. Hypo is less. What is less? Solute is less as compared to cell sap and other solution. Hyper is more. What is more? Solute is more as compared to cell sap or another solution. ISO is equal. What is equal? Solute concentration as compared to cell sap or another solution. End osmosis is inward movement. कब होता है? When you place it in a hypotonic solution. X osmosis is outward movement. कब होता है? When you place it in a hypertonic solution. ये tonicity का definition है textbook का. Relative solute concentration of solution that determines direction and extent of diffusion. ये वाला एक बार ही check कर लेना. थोड़ा confusing है, but that's the definition that they've given you. You can remember relative concentrations of solutions. That is tonicity. फिर डायग्राम बेस्ड क्वेश्चन का बहुत स्ट्रॉन्ग कंटेंडर है प्लाज्मोलाइसिस का क्वेश्चन. प्लाज्मोलाइसिस के क्वेश्चन में हाउ डू यू आइडेंटिफाई? सेल वॉल विल रिमेन एज इट इज. Cell membrane will shrink away from the cell wall. When the cell membrane will shrink away from the cell wall, the space between the cell wall and the cell membrane will be filled with hypertonic solution. The cell membrane will shrink, the vacuole will reduce in size, and you will have the plastids and the nucleus inside. Plasmolysis will be contraction of the cell contents along with the cell membrane away from the cell wall when placed in a hypertonic solution. If they give you the plasmolysed cell, they will ask you in what kind of solution was the cell placed? Hypertonic. What can you do to get the cell back to its original condition? Place it in hypotonic solution or pure water. Which kind of osmosis will happen when you are bringing it back to its original condition? End osmosis. What kind of osmosis resulted in the cell becoming plasmolysed? Exosmosis. You could, if they ask you the state, then you could have answers like flaccid, or you could have answers like plasmolysed. So that's the plasmolysis wala. See, this was a board question. Space between the cell wall and the cell membrane in a plasmolysed cell is filled with what kind of solution? So hypertonic solution. State could be plasmolysed. In case there's no plasmolysed over there, look for flaccid. Process is plasmolysis, which is basically the shrinkage of the cell contents along with the cell membrane away from the cell wall when placed in a hypertonic solution. Then see deplasmolysis, return to an original condition was deplasmolysis. Move down. This is the diagram that we are looking at. See this kind of diagram. Don't get confused by these multiple layers over here. These are still cell wall only. Okay, that's still the cell wall that you are looking at. So that's the cell wall, multi-layered or thick cell wall. The shrunken part away, this part over here, this is your cell membrane, which you see as 3 over there, I think. Yeah, that's the cell membrane. So here they'd ask what is the state. So we can write plasmolysed. Name the structure that acts as a selectively permeable membrane. So cell membrane. Part numbered 1 to 4. 1 obviously is the cell wall. 2 is the space between the cell wall and the cell membrane, which is hypertonic solution in case it is a plasmolysed cell. 3 is the cell membrane and 4 is the nucleus. How can you get it back to its original condition? You place it in a hypotonic solution. Hypotonic solution में रखो या pure water में रखो. And scientific term for recovery is deplasmolysis. So scientific term for recovery is deplasmolysis. How does recovery take place? Because water goes inside. Okay, water goes inside. So when water goes inside, it's basically endosmosis that we are looking at. 2 features of plant cell which is not present in animal cell. You could look at plastid. Okay, you could look at cell wall. Plastid and cell wall is what you could look at. Similar questions on plasmolysis are in the workbook. You can refer to them. Then your diagram of root hair. This one is a turgid root hair, the one which is filled with water. You can see this is the cell wall here. This is the cell membrane. This is the nucleus. Okay, this is the idea. That's a fill in the blank question. Unicellular extension. Root hair unicellular होता है वो वाला concept. तो root hair is unicellular. दूसरा concept है वो रूट हेयर का सेल सैप हाइपरटोनिक होता है, ये यहाँ पे मार्क किया है, सेल सैप ऑफ रूट हेयर इज हाइपरटोनिक। परमिएबिलिटी वाला डिस्टिंग्विश हमने ऑलरेडी देख लिया है। When they ask you to distinguish on the basis of hyper and hypotonic, when they ask you to distinguish on the basis of permeability, it is freely permeable, semi-permeable. If they ask you on basis of composition, then cell wall is cellulose, cell membrane is lipoprotein. When this is a plasmolized root hair, we had shown कि plasmolized नहीं पूछेगा तो क्या पूछ सकता है कि the root hair diagram when excess fertilizer is added to it, क्योंकि excess fertilizer becomes hypertonic and hyper में रखेगा तो X होता है। तो here you see that there is a drooping structure, ये पूरा shrink हो गया है। अगेन ये वाला सेल वॉल है, ये वाला सेल मेम्ब्रेन है, और ये जो अंदर का सोल्यूशन है यहाँ पे, ये हाइपरटोनिक सोल्यूशन है। देन टर्गर एंड वॉल प्रेशर, टर्गर प्रेशर इज अंदर से बाहर बिकॉज ऑफ द सेल कंटेंट्स ऑन द सेल वॉल इन अ फुल्ली टर्जिड कंडीशन। वॉल इज बाहर से अंदर, बाय द वॉल ऑन द सेल कंटेंट्स। अगर कंफ्यूजन होता है तो वॉल से वॉल आता है, सिंपल। वॉल प्रेशर वॉल से आता है, तो अगर वॉल प्रेशर वॉल से आता है तो टर्गर प्रेशर इसका ऑपोजिट है। तो किससे आएगा? सेल कंटेंट से आएगा ऑन द सेल वॉल। जब सेल फुल्ली टर्जिड होगा तो टर्गर प्रेशर विल बी इक्वल टू वॉल प्रेशर। ओके, अगर सेल फुल्ली टर्जिड है, टर्जिड मतलब फुल वाटर से भरा हुआ है, तो वो कंडीशन में टर्गर प्रेशर विल बैलेंस द वॉल प्रेशर। और एल्स if the turgor pressure increases more than the wall pressure, then the cell will burst, which is a board MCQ question. It's already marked important. Under what conditions of turgor and wall pressure does the cell burst? So the cell will burst when the turgor pressure increases more than the wall pressure. When is the cell turgid? The cell is turgid when the turgor pressure balances the wall pressure. So we had seen that the definitions are again important. Then we had seen the idea of एक और plasmolized cell था. The give reasons. Roots of plant wilt when excess fertilizer is added because fertilizer acts as a hypertonic solution, जिसके वजह से X osmosis होता है, plasmolysis होता है। ये marking points लिखे थे यहाँ पे, hyper, X and plasmolysis. Second वाला give reason देखा था, salt is added on weeds to kill them because salt again is hypertonic. Weeds are unwanted plants that grow in the fields. उनके ऊपर salt डालोगे तो salt will act as a hypertonic solution. उनमें से पानी बाहर आएगा by X osmosis leading to plasmolysis. We gargle with salt water in case of throat infection. Again, salt water is hypertonic. जब आप gargle करोगे तो वो जो बैक्टीरिया जिसने throat infection cause किया है उससे X osmosis होके plasmolysis होगा. The bacteria will get killed. Same idea. Fifth one now. Freshwater fish cannot survive in salt water क्योंकि salt water hypertonic होता है. Freshwater fish को अगर रखोगे उसमें तो X osmosis हो जाएगा, water will come out. Chefs prefer adding salt to salad just before serving it. The marking point was just, क्योंकि अगर पहले से डाल के रखोगे तो क्योंकि salt removes water outside the vegetables, it will become soggy, it will become flaccid. Potato cubes placed in water become firm and increase in size. Again, a board question. Becoming firm, always remember, is with turgid, and increasing in size मतलब water is entering inside, मतलब end osmosis. So water acts as a hypotonic solution and the incoming water exerts a turgor pressure, which is what results for the firmness. Marine fish burst. This was also there in the sample paper. Marine fish bursts when placed in tap water. कोई चीज अगर burst हो रही है मतलब उसके अंदर बहुत कुछ जा रहा है. So that's basically end osmosis की वजह से burst हो रही है. अगर कोई चीज burst नहीं हो रही है, if something is losing water, then it will not burst. Something has to go inside it, then it will burst. So the marine fish basically has salt in its body, due to which it becomes hypertonic, and when you place it in water, end osmosis takes place, and when end osmosis takes place, this structure will rupture. Then we moved on to seeing the concept of root pressure. Leaves of Mimosa pudica wala. Leaves of Mimosa pudica mein the marking word you have to remember, marking term you have to remember is changes in turgor pressure in the pulvinus. That's it. Pulvinus is the swollen leaf base, and it is able to detect changes in turgor pressure. Then we saw the idea of root pressure. This was the diagram based question. Again, another expected diagram based question candidate. Soil water, root hair, cortex, endodermis, xylem is the logical sequence. Root hair ka identification is not a problem. The identification problem is ये region, this cortex. Okay, this single with thickened is the endodermis. उसके बाद pericycle होता है, but pericycle I doubt they will ask. But this is the xylem vessel. Identifying the xylem vessel should be easy. Then you remember root pressure के लिए alternate turgidity, flaccidity, continuous inward movement of water leading to end में ascent of sap. So one of the factors responsible for ascent of sap is root pressure. Root pressure is the pressure developed in the roots due to continued inward movement of water leading to alternate turgidity and flaccidity and contributing to ascent of sap. Root pressure excess होगा तो guttation होता है, which is the loss of water in the form of water droplets from hydatodes located at margins of the leaves. So root pressure का logical sequence soil water के बाद root hair के बाद cortex के बाद endodermis के बाद xylem. How does water enter the root? So water enters the root by endosmosis or osmosis also. How does, again, if you have a problem with understanding endosmosis, osmosis, don't worry. They will not give you such overlapping ones. In case they do and you still want to distinguish, then remember, in case they do and they still, you still want to distinguish, then remember if there's a living membrane, जैसे cell दिया हुआ है, तो उसमें endosmosis use करना है. अगर parchment paper दिया हुआ है, तो उसमें osmosis use करना है. Living membrane के through osmosis, living membrane रहेगा तो you can write के through when it is there, you can specify endosmosis, exosmosis. But they will not give you confusing questions, so do not worry. How does water move in the cortical cells? It moves from one cell to another by cell to cell osmosis. The root pressure का definition, उसके बाद remembering that it helps in ascent of sap and remembering that उसके excess से guttation होता है. ये बड़ा आपका board question 2016 वाला. So you have the root hair, then you have the soil particles. 4 is the cortical cells. बस इतना ही पूछा था. As I told you, बीच वाला they will not ask, and this is the xylem. So this was the 2016 question that was there. Then what is ascent of sap? So ascent basically is rise, and sap. Sap is water and minerals. कहाँ water and minerals? Water बोला मतलब obviously xylem. So ascent of sap takes place in the xylem. So it's the upward movement of water and minerals in the xylem. बहुत सारी चीजों की वजह से ascent of sap होता है, जिसमें से एक root pressure होता है जो अभी हमने discuss किया. दूसरा transpirational pull होता है, which is that suctional force which is created due to transpiration. Suctional force which is created due to transpiration. तीसरा होता है adhesion cohesion. You can see adhesion cohesion का image you can see. दो water molecules के बीच का जो force of attraction होता है वो cohesion होता है. Cohesion is force of attraction between similar molecules, and adhesion is force of attraction between dissimilar molecules. So dissimilar molecules will be walls of xylem and water molecules, and similar molecules will be water and water molecules. So adhesion and cohesion. Adhesion is force of attraction between dissimilar molecules. Walls of xylem and water के बीच का जो attraction होता है वो adhesion होता है. And water के बीच का जो attraction होता है वो cohesion होता है. So cohesion की वजह से water molecules can maintain one continuous column. Very commonly asked MCQ that you've been seeing doing the rounds. Then you have your capillarity. The thinner the tube, the greater the rise. Xylem vessels are narrow. क्योंकि वो narrow रहता है इसलिए water can rise up to greater heights. Then girdling experiment में कैसे पता करना है कि क्या निकाला हुआ है और क्या है. अगर लीव्स ड्रूप कर रहा है मतलब जाइलम नहीं है. अगर लीव्स टर्जिड है मतलब जाइलम है. लीव्स ड्रूप कर रहा है तो जाइलम नहीं है. जाइलम नहीं है तो क्या है? फ्लोएम है. अगर लीव्स टर्जिड है तो जाइलम है. अगर जाइलम है तो क्या नहीं है? फ्लोएम नहीं है. okay, रिमेंबर टर्जिड लीव्स मतलब जाइलम है, और फ्लैसिड या ड्रूपिंग लीव जैसे यहाँ पे लीव्स विल्ट कर रहे हैं तो ये वाले में जाइलम नहीं है, और ये वाले में टर्जिड है लीफ मतलब ये वाले में जाइलम है. जाइलम क्या करता है? वाटर ट्रांसपोर्ट करता है अलोंग विथ मिनरल्स. कहाँ से? रूट से. कहाँ पे? अपवर्ड डायरेक्शन में टू ऑल द अदर प्लांट पार्ट्स. फॉर फ्लोएम फूड. फर से फूड. सो फ्लोएम फूड ट्रांसपोर्ट करता है जहां से बनता है, विच इज बेसिक द लीव्स टू अदर पार्ट्स ऑफ द प्लांट इन अपवर्ड एज वेल एज डाउनवर्ड डायरेक्शन. और जाइलम एंड फ्लोएम को साथ में वैस्कुलर बंडल बोलते हैं. Okay, so you see which one is removed. That is how you will understand girdling experiment. फिर osmotic pressure में pressure which is preventing osmosis. Pressure which is preventing osmosis. Remember the pressure which is preventing osmosis. Osmosis के setup में अगर piston दिखेगा तो वो osmotic pressure के लिए measure कर रहा है. बस और कुछ नहीं करना है. Definition के अलावा there is no other understanding of osmotic pressure you need. फिर imbibition. दो गिव रीजंस. वुडन डोर्स एंड ड्रॉर्स आर डिफिकल्ट टू क्लोज इन रेनी सीजन वाला, क्योंकि वुड बेसिकली इज हाइड्रोफिलिक Hydrophilic matlab water loving. So whenever the moisture content is high, it absorbs water. Water is on the surface. And because of that, the size is going to increase. It also exerts a pressure, which is imbibitional pressure, due to which a can of seeds may burst when water accidentally enters inside it. तो ये can of seeds वाला give reason आता है, और दूसरा wooden doors and drawers are difficult to close in rainy season. So imbibition is the process in which living or dead plant cells in their dry or semi-dry state absorb water by surface attraction. Most of the definitions will have surface attraction, so look for surface attraction. So wood or cellulose is hydrophilic, which means it's water loving. The process by which, that's the definition. Ye wala tha, sorry, I missed this one. Significance of imbibition was rupture of the seed coat. The seed coat rupturing during the process of germination is by imbibition. That was the imbibition concept that we spoke about. I think absorption by roots mein itna hi tha.